Imagine laughing at a joke or just sneezing and instantly panicking.
Right, panicking about where the absolute nearest bathroom is.
Exactly. And the thing is, up to a fourth of grown women don't actually have to imagine that. I mean, it
Yeah.
just, their daily reality.
Yeah, but because it involves intimate anatomy, it's shrouded in almost complete silence.
It really is. We are talking about stress urinary incontinence, or SUI. It's this massive hidden epidemic of discomfort that causes you to, alter your lifestyle, give up exercise, and just suffer an embarrassment.
And that heavy silence makes it incredibly difficult to figure out what actually works if you finally decide to seek help.
Which is exactly why we compiled today's stack of sources for this deep dive. We're looking at recent clinical meta-analyses on regenerative medicine from Frontiers and MDPI, alongside some pretty major regulatory updates directly from the FDA and the UK's MHRA.
Right, and to contrast all that rigorous science, we're also looking at a big pile of marketing materials from high-end aesthetic clinics.
Yeah, it's quite a mix. Our mission today is to figure out if a cutting-edge biological phenomenon called exosomes, which, are currently being aggressively marketed as intimate rejuvenation, can actually cure SUI.
Or if this is all just premature gray market hype taking advantage of desperate patients.
Right, because before we can understand why exosomes are being hailed as this new frontier, we really need to understand the mechanical failure of the current treatments. I mean, if we look at the mechanics of SUI, what is actually breaking down in the body?
Well, at its core, SUI is a mechanical failure of the urinary sphincter and the pelvic floor. So the pathophysiology usually involves two main issues.
Okay.
The first is a weakened pelvic floor, which is often due to the trauma of childbirth or just age-related tissue degradation. This leads to what's called urethral hypermobility.
Meaning things are moving around too much.
Exactly. The urethra literally loses its structural support and kind of shifts out of place. And then the second issue is intrinsic sphincter deficiency.
So, meaning the actual muscle valve that's supposed to hold the urine back just isn't strong enough anymore.
Right. It can't withstand sudden pressure spikes in the abdomen.
Ah.
That pressure spike is the key.
What?
Like when you cough, jump, or lift something heavy, your intra-abdominal pressure just shoots up.
And in a healthy system, the support structures absorb that shock.
Yeah, and the sphincter stays clamped shut. But when the support is compromised or the valve is weak, that pressure forces urine right past the barrier.
Wow. So for moderate to severe cases where, lifestyle changes or pelvic floor apps aren't enough, doctors have historically pointed patients towards surgery.
They have, yeah.
And for years, the gold standard fix has been the midurethral sling, or MUS, typically made of a synthetic polypropylene mesh. But putting a synthetic mesh sling in there to hold up the urethra, it feels like putting a plastic hammock under a leaky pipe.
That is a perfect analogy.
Because it provides purely mechanical support, right? Holding things in place, but it does absolutely nothing to heal the underlying tissue or fix the broken valve.
That plastic hammock analogy gets right to the heart of the failure. I mean, the human body is this dynamic living environment.
Right.
Highly sensitive, innervated vaginal tissue violently rejects rigid, inert synthetic plastics. The mechanics of that rejection are just brutal.
Because the rigid plastic creates friction.
Exactly. Friction against the soft moving tissue, which causes micro tears.
Which sets off the body's alarm bells, I'd imagine. Like, the immune system sees this permanent plastic mesh as a foreign invader.
And it completely attacks it. Immune cells called macrophages rush in, trying to break down a plasma they can literally never dissolve.
Oh, wow. So it just stays inflamed.
Yeah, this leads to chronic, severe inflammation. The body eventually tries to just wall off the mesh by encapsulating it in dense, rigid scar tissue.
That sounds awful.
It is. And for a significant subset of women, this cascading reaction leads to severe complications. We see mesh erosion, where the plastic actually rubs entirely through the vaginal wall.
Oh my God.
Or it even cuts into the urethra itself.
The sources outline how debilitating this is. I mean, we're talking chronic pelvic pain, recurrent infections, agonizing revision surgeries. It got so bad that regulatory bodies had to essentially pull the emergency brake.
They really did. The UK's National Institute for Health and Care Excellence, NICE, explicitly stated that due to these massive safety concerns, transvaginal mesh for prolapse should now only be used in the context of rigorous research.
Wow. So it's no longer a standard treatment option.
No, not at all.
So if the plastic hammock fails and actually harms the surrounding structures, we need a way to rebuild the pipe itself, right?
Yeah.
We have to regenerate the tissue.
That's the logical next step.
And scientists first looked at stem cells.
Yeah.
Harvest the body's master builders, maybe from a patient's own fat or bone marrow, and inject them into the damaged sphincter to grow new muscle.
The theory was sound. You have a depleted muscle, you add cells capable of becoming new muscle.
Right, but we have a recent 2026 meta-analysis from Frontiers in Medicine looking exactly at this autologous stem cell therapy for female SUI. They reviewed 19 studies with hundreds of patients. And, okay, let's unpack this. The data shows a pooled cure rate of 41% and a pooled improvement rate of 55%.
Yeah.
I'm not going to lie, a 31% cure rate doesn't sound like the regenerative miracle we're looking for.
No, the clinical results were undeniably underwhelming.
And beyond the mediocre success rate, you're still injecting live, dividing cells. Aren't we worried about the inherent risks of live cells? Like immune rejection or the cells mutating and multiplying out of control into tumor?
Oh, absolutely. The literature validates all of those concerns. Live stem cell therapy presents a logistical and biological nightmare. There's the risk of tumorigenicity.
Right.
Plus the intense difficulty of keeping the cells alive outside the body, expanding them in a lab, and then hoping they survive the injection.
But wait, why wouldn't they survive? I mean, they're designed to heal, aren't they?
Well, because you are dropping them into a hostile wasteland. When the pelvic floor undergoes trauma and chronic inflammation, the tissue becomes fibrotic.
Meaning scar tissue forms.
Exactly. And scar tissue lacks a robust blood supply. So you're taking expensive, delicate, laboratory-nurtured stem cells and injecting them into a tough, oxygen-starved environment.
So they just die.
Most of the injected stem cells simply die within days.
Okay, so if the vast majority of the stem cells are dying almost immediately in that harsh environment, how are 41% of the patients still getting cured?
What's fascinating here is that scientists realize the cells themselves aren't actually integrating and becoming new tissue.
Wait, really?
Yeah. The stem cells aren't moving into the neighborhood and building houses. The healing comes from their sweat.
Okay, you're going to have to explain stem cell sweat to me.
So the scientific term is the secretome. While those stem cells are alive, even for just a few days, they're desperately pumping out chemical signals into their environment.
Trying to fix things before they die.
Pretty much. They release these signals to promote healing, reduce inflammation, and basically stimulate the local resident cells to wake up and repair the damage. The realization was profound. We realized we don't actually need the stem cell factory itself. We just need the packages they're shipping out.
And those packages are the exosomes.
Bingo! Exosomes are incredibly tiny extracellular vesicles. We're talking nano-sized, about 30 to 150 nanometers in diameter.
That is tiny.
They're essentially microscopic lipid envelopes released by the cells.
So they're basically the body's biological text messages.
That is the perfect way to visualize them. And the key here is they contain absolutely no live cells.
Which completely eliminates the risk of them turning into a tumor.
Zero tumorigenic risk. But they are packed to the brim with messenger RNA, microRNA, and potent proteins. For example, they carry a protein called CXCL12.
Let me guess, that acts like a biological flare gun. It fires off a signal that recruits the body's native repair cells to the exact site of the injury.
Yes. And that flare gun is highly specific. When CXCL12 goes off, it pulls circulating stem cells directly to the urethral sphincter.
That's incredible.
They also carry BDNF, which is a protein that specifically directs the regeneration of damaged nerve tissue.
Okay.
So when you inject these exosomes, they text the local cells with specific instructions. They say, turn off the inflammation pathway, stop laying down rigid scar tissue, meaning they're highly antifibrotic, and start growing new blood vessels to bring oxygen back to the area.
That is wild. Instead of trying to transplant foreign cells and hoping they survive the oxygen-starved wasteland, you just isolate the chemical text messages that instruct your own cells to fix the environment.
Exactly.
It solves the safety issue, and I imagine a vial of microscopic envelopes is vastly more stable to manufacture and store than living tissue.
It fundamentally changes the entire supply chain of regenerative medicine.
I bet. But, theory is great, but we need to see it actually work in the body.
Yeah.
How do these biological text messages perform when they're actually delivered to a damaged urinary sphincter?
Well, the preclinical trials are where this gets genuinely exciting. The sources detail animal models that simulate the specific types of childbirth injuries that lead to SUI in humans.
Right, and one study really stood out to me. Researchers took exosomes that were secreted by human urine-derived stem cells, and they injected them into rats with simulated pubococcygeus muscle injuries.
Yeah.
And that's the main hammock muscle of the pelvic floor that supports the urethra. Right.
That's the one.
And the exosomes successfully promoted the repair of that specific muscle damage.
They did. And the research scales up beautifully to larger animal models, too, which anatomically mirror human structures much more closely.
Like the pig study.
Exactly. There's a pivotal study utilizing a porcine model. Researchers created a hydrogel out of collagen and combined it with platelet-derived exosomes.
Wait, okay, if exosomes were only 30 nanometers across, I imagine if you just inject them in liquid saline, they'd immediately get washed away by the bloodstream before they could even deliver their message. Is that what the collagen hydrogel is for? Like a sticky trap?
You've hit the nail on the head. The hydrogel acts as a localized, sticky scaffolding. It holds the exosomes exactly at the urethral sphincter so they have time to interact with the damaged tissue.
Oh, that's clever.
And the results were astounding. Injecting this exosome-loaded hydrogel completely restored the damaged urethral sphincter function at 7 weeks.
Wow.
And just to isolate the variable, they injected a control group with just the collagen hydrogel.
Let me guess. It did nothing.
It did absolutely nothing. The repair was entirely driven by the exosome text messages.
See, here's where it gets really interesting, though. Because it's not just about fixing the original childbirth injury. One of the sources highlights a study where exosomes were used to heal vaginal tissue that had been damaged by previous mesh exposure.
Yes. The very complication caused by the synthetic slings we discussed earlier.
Think about the magnitude of that. Instead of just patching a leaky pipe with a plastic hammock, we're texting your body's natural construction workers to come in and rebuild the floor entirely. Right. And on top of that, we're using those same biological text messages to clean up the toxic, scarred mess left behind by the failing plastic mesh.
Yeah, and if we connect this to the bigger picture, it really highlights how exosomes literally reprogram the extracellular matrix.
The extracellular matrix being like the structural scaffolding that holds all our cells together.
Correct. After an injury, that scaffolding gets warped by stiff scar tissue. But by delivering targeted cargo, like a specific strand of RNA called microRNA 93, the exosomes force the tissue to break down that rigid scarring.
And lay down healthy, flexible collagen again.
Exactly. They wake up dormant satellite cells to rebuild muscle fibers. It is a true regenerative cure aimed at restoring the anatomy to its pre-injury state.
I mean, hearing all this, you're probably thinking, sign me up, where do I get this?
Right. If exosomes can rebuild a pelvic floor in the lab and fix mesh damage, can you go get this treatment today?
Well,
Because if you look online, the answer seems to be a resounding yes. But our sources make it very clear that there is a dangerous catch, a massive catch. When you leave the realm of peer-reviewed animal models and enter the consumer market, you cross into the Wild West.
yeah, we reviewed marketing materials from several high-end aesthetic clinics. If you scroll through the websites of places in London or Los Angeles clinics like Symmetry Aesthetics or Bauer Medical, you aren't seeing cautious clinical language.
Not at all.
You're seeing slick, aggressive marketing for intimate health. They brand these procedures. You'll see trademarks for things like the Bailwash shot right next to the Botox. They frame it as vaginal rejuvenation, which really taps into a very real desperation.
And the marketing is incredibly effective. According to Top Doctor magazine, consumer search interest in exosome therapy has surged 557% year over year.
That is a massive jump.
Patients want a cure, and these clinics are using the real underlying science we just discussed to sell immediate access to products.
So what does this all mean? If a listener sees an ad for a $5,000 exosome intimate injection to cure your incontinence, is it legitimate? Are you actually getting those miraculous little text messages?
This raises an important question about regulatory reality, and the answer is pretty blunt. As of 2026, there are 0 FDA-approved exosome products for therapeutic use in humans.
0.
None. And the MHRA in the UK holds the exact same stance. They have 0 licensed exosome products for SUI, alopecia, or intimate rejuvenation.
Okay, wait. If there are 0 approved products, how are these clinics operating right out in the open?
They rely on a legal gymnastics routine known as the topical loophole.
The topical loophole.
Yeah. The FDA and MHRA rules generally state that if a product is strictly a cosmetic, meaning it's only meant to be applied to the surface of the skin to cleanse or beautify without altering the body's underlying biology, it doesn't require rigorous pre-market clinical trials.
Okay.
So manufacturers label these vials of exosomes as topical cosmetic serums.
But no one is paying $5,000 to just rub a serum on the outside of their skin for a severe internal muscle deficiency. The clinics are loading it into syringes and injecting it directly into the pelvic floor.
Exactly. And the moment a clinic claims the product will regenerate tissue, stimulate muscle, or cure SUI, or the moment they puncture the skin to inject it, that product legally becomes an unapproved, illegal biologic drug.
Wow.
The FDA has actually issued multiple public safety notifications and warning letters targeting these exact practices.
And the contents of those warning letters are terrifying.
Yeah.
I mean, we aren't just talking about a bureaucratic slap on the wrist for mislabeling. We're talking about severe threats to patient safety.
Yes. The FDA cites unvalidated sterility. Because these gray market products bypass the clinical trial approval process, nobody is independently verifying the manufacturing standards.
That's sketchy.
Vials have literally been found contaminated with bacteria or endotoxins.
And endotoxins are basically the toxic shells of dead bacteria, right? So if you inject those into a human body, it can trigger a massive immune shock.
It can cause severe systemic reactions, yeah. The warning letters also cite unknown cell lines.
Meaning you don't know where it came from?
You don't even know exactly what type of tissue these exosomes were harvested from or how the donor was screened.
That's crazy. You are paying thousands of dollars out of pocket with zero insurance coverage and zero regulatory protection if you develop a severe infection or a life-threatening allergic reaction.
Which is why we really have to talk about the green flags. If you want access to this cutting-edge science right now safely, how do you actually navigate this?
Right. What should people look for?
There is only one legitimate, safe pathway today. You must ensure the treatment is part of an FDA-cleared clinical trial. The clinic or institution must be able to provide you with an active IND number.
IND.
That stands for Investigational New Drug. The trial should be overseen by an institutional review board, and it's usually affiliated with a major research university or hospital.
So if a glossy aesthetic clinic guarantees a cure, pressures you to buy a package today, and can't produce IND documentation
Run the other way. The preclinical science is revolutionary, but the commercial market is currently predatory.
Man, it is a lot to process. We've traced the mechanical failures of synthetic plastic mesh, discovered the biological promise of stem cell sweat, and crashed headfirst into a regulatory gray market. Let's bring this all together.
Yeah.
Exosomes hold massive science-backed potential. They really could change the treatment of stress urinary incontinence from a dangerous mechanical scaffolding to genuine cellular-level regeneration.
But right now, today, patients are caught in the treacherous gap between incredible lab results and predatory clinics.
It's the classic trajectory of a medical breakthrough, really. The basic science accelerates faster than the safety regulations can keep up, and commercial opportunists just rush in to fill the void.
But the destination, a true regenerative cure for SUI, is clearly visible on the horizon.
And thinking about that horizon brings up a pretty wild thought. Right now, to get exosomes, we have to harvest them from human stem cells in a lab, which is, expensive, difficult to standardize, and carries the risk of using human donor tissue. But since exosomes are essentially just microscopic lipid envelopes carrying chemical software like RNA and proteins, what happens when we no longer need human cells to manufacture them?
Oh, that's an interesting question.
Right. Like, could we one day computationally design and 3D print synthetic exosomes? Imagine a perfectly sterile lab-made envelope, loaded with bespoke RNA instructions tailored perfectly to your specific biology,
That would be amazing.
offering a customized software update to completely cure your unique incontinence.
The field of synthetic biology is already moving rapidly in that direction, actually. If we can master the creation of the payload and the delivery vehicle from scratch, we remove the unpredictability and contamination risks of human cell lines entirely.
It is amazing to think about. From the stigma and silence of suffering with a leaky pipe to the failed, painful attempts of holding it up with a plastic hammock, we are finally learning how to speak the body's own cellular language to rebuild the plumbing entirely.
We really are.
You are now armed with the knowledge to navigate this wild frontier of medicine. Stay curious, ask the hard questions, and don't let anyone sell you a topical serum in a syringe without the clinical data to back it up. We'll catch you on the next deep dive.